We'll start by learning 4 important things. First, this is a chromosome and this is also a chromosome. You can see here there's a centromere attached to one chromatid. This is another centromere attached to 2 chromatids, and both of these are considered chromosomes. So, as long as there is at least one chromatid attached to the centromere, both of these structures are considered to be chromosomes. Okay? Then the DNA content of a cell is represented by the letter C. Okay? And the number of complete sets of chromosomes is referred to as N, small letter N. In one cell, when we are writing about the amount of DNA content or chromosomes, we mention it as 2N and 2C. And this holds true for diploid cells. Okay, all of the cells in our body except our gametes are diploid cells, and we've discussed about ploidy in Embryology Basics 1. You can refer that video. So in cells whose ploidy is 2, that is diploid cells, we write the content to be 2N and 2C, and every cell has 46 chromosomes or 23 pairs of chromosomes. Now ploidy is, we have all of these chromosomes in pairs, so they are in sets of 2. That means ploidy is 2. Now before the cell enters meiosis, it has to undergo interphase. Okay, so the cell cycle, as you've seen in the Embryology Basics 2 video, the cell cycle begins with interphase, which is G1, S and G2, followed by your meiosis or mitosis. So here in meiosis we have meiosis 1 and meiosis 2, but anyways, to begin with, it has to undergo G1, S and G2. So in G1, what happens? Now here, like in the previous video that I drew for mitosis, I'm going to be drawing just 2 chromosomes to represent the whole set of chromosomes. So we have 2 chromosomes here, they have 1 chromatid each. So this is in G1. This is a diploid cell. We are starting with a diploid cell, so the content is 2N and 2C. In S phase, there's DNA replication, meaning number of chromosomes are same. See, we just saw that this is also a chromosome, so the number of chromosomes is going to be the same, but the amount of DNA has multiplied. It has replicated by 2. So we have 4C amount of DNA. Now in G2, the same thing, so 2N and 4C, no change. Now coming to prophase 1, which is meiosis 1's first phase. Okay, so prophase is made up of leptotene, zygotene, pachytene, diplotene and diakinesis. Now leptotene is just chromosomal condensation. In zygotene, homologous chromosomes are arranged next to each other. In pachytene, they'll be crossing over, and then diplotene, they'll separate and they form chiasmata, and then diakinesis, then they enter metaphase. So here nothing changes. The structure of the chromosome, only the orientation and the way they are attached with each other is going to change. Okay, so here we saw there are separate, the chromosomes are separate. Here they are joined together by their chromatids, non-sister chromatids. In diplotene, what happens? They are separated at all points except at the place where crossing over took place. So this structure is called a chiasmata. Okay, this is called a tetrad. This is zygotene actually. These are homologous chromosomes. So from a tetrad or a bivalent, it becomes a chiasmata in diplotene. Okay, so the number of chromosomes or the DNA content does not change. So 2N4C holds true throughout prophase 1. Nothing changes. In metaphase, it's the same thing. Again, 2N and 4C. See, metaphase, anaphase, telophase, they're going to be the same as they were in mitosis. Okay, the concept of metaphase, anaphase, telophase remains the same. The only difference is here we have an X-shaped structure because of the chiasmata. So metaphase, nothing changes, 2N and 4C. Anaphase, what happens? We have 2 centrosomes. They're attached to the kinetochores via mitotic spindles, and they pull these chromosomes apart. So the number of chromosomes here is the same. Okay, we started with 2N number of chromosomes, that has right? 2n number of chromosomes, and that has been true so long. Nothing has changed. Now, why are we not calling this one chromosome? Even though they are attached together, why are we not considering this entire thing to be one chromosome? Because they have different centromeres and they're only attached here because of the crossing over, okay? So that is not one chromosome. These are still 2 chromosomes, so the number has not changed at all. The same thing again. And the genetic material, of course, the content has not changed, so 4C. In telophase, what happens? They are separated and the nuclear envelope begins to form in preparation of cytokinesis so that 2 daughter cells can form. Still nothing has changed, so 2n and 4C. Okay, so in meiosis 1, okay, this is meiosis 1, we started with 2n and 2C and we are ending with 2 daughter cells, okay? And the total amount here before the daughter cells divide, before the daughter cells are produced, is 2n and 4C. So when this divides, what will happen? This information, this is the total content, whatever is there in the cell is this number, is the set of numbers 2n and 4C. So when you divide it by half, what happens? 1n, 2C, 1n, 2C. So we started with the parent cell which was diploid, 2n and 2C amount of information, and we are finishing meiosis 1 with 1n and 2C amount of information, okay? So each daughter cell will have 1n and 2C. So like I said before, this has 1n and 2C amount of information. This also has 1n and 2C amount of information. These are daughter cells from meiosis 1, okay? Now we're going to start prophase 2, okay? So from meiosis 1, it's entering meiosis 2. Now in meiosis 1, before the cell started meiosis 1, it had to go through interphase. But here, nothing of that sort, okay? Since it went through interphase, the amount of genetic material got replicated. But now, nothing is getting replicated. You are going to use the same 1n and 2c amount of information to get through meiosis 2. Now, meiosis 2 is very similar to mitosis. It's almost all the same things except for the kind of chromosomes that are getting divided, okay? So, we have 1 and 2c. Prophase, nothing, it's just visible chromosomal condensation. So it's the same thing, 1n, 2c, 1n, 2c, okay. Metaphase, same thing, they are aligned along the metaphase plate, that's the only difference. So 1n, 2c, 1n, 2c. Anaphase, what happens? These chromosomes in metaphase, they'll be pulled apart by the spindles. You already know that. So now the number of chromosomes has doubled. Each chromosome that existed previously here has been halved. So even though the amount of genetic material is the same, which is 2C and 2C, the number of chromosomes is increasing, okay? So if that was a chromosome, this is also a chromosome. Now here we had only 1 chromosome, now we have 2. So it has doubled. So we have 2n and 2n. Now it has become 2n and 2c per cell. Telophase, the same thing, just nuclear envelope starts to form, nuclear division is complete. So 2n, 2c, 2n, 2c. Then the cells undergo cytokinesis. This cell has given these 2 daughter cells, this has given these 2 daughter cells. Now 2n and 2c was the total content. We are going to divide this by 2. So 1n, 1c. 1n, 1c, 1n, 1c, and 1n and 1c. So we have 4 daughter cells which are haploid. Diploid was 2n and 2c, which was the parent cell, okay? And haploid are these 4 daughter cells. That is 1n, 1c, 1n, 1c, 1n, 1c. Yeah. So meiosis is what the gametes undergo, because gametes have to be haploid in order for them to fuse together and form a diploid cell. So gametes undergo meiosis so that they can convert from a diploid cell to a haploid. That is, 1 diploid cell will give rise to 4 haploid daughter cells. Now remember, even if you forget everything that I just said, please try to remember these 4 points, because if you know these things, you can calculate all of this all by yourself. This is just common sense. So these 4 points are important. Remember, the content is referred to as C. The amount of genetic material, the DNA, is C, and the number of complete sets of chromosomes is N. And we have 46 chromosomes or 23 pairs. And whether it has 1 chromatid or 2, it is still considered a chromosome. Okay.